amino acids. Uh, Proteins are gonna be, they are a key component in many things, but one of the most important that they serve, other than structure, is the fact that they are the receptors, the identifiers for cells to see that they are your cells. And remember, every one of us, we may have a protein that does the same thing, but it's gonna be in our receptor cells. They all may act as receptors, but they have a different makeup. And by that makeup, then it changes their shape. So cells such as your white blood cells going through your body all the time, out into the fluids outside of the muscles and in the cell, they're scanning. They're just like a scanner at the supermarket. If they find a cell that has membrane proteins that they do not recognize, then they stimulate the immune response to come and remove those cells, kill them. So they are very important immunologically. They make up structure as we see it. They are enzymes. And remember, the things I'm asking you right now, folks, are the things you'll see on the thing. Proteins end, the ending of when you see a protein, it will end in IN. Insulin ends in IN. That tells you it's protein. If it ends in ASE, it is an enzyme. Normally, an enzyme is named after what it works on. If it is an enzyme to break down lactose, then the name becomes lactase. So keep in mind those things. Those are the types of questions, along with what is the primary structure, the secondary structure, the tertiary, and the quaternary structures of proteins. Make sure you understand how to identify those. I try to keep the part on proteins and this particular unit straightforward and simple. But you do need to basically know those definitions and what makes a tertiary. Remember, primary, single ribbon, just the chain of amino acids. Secondary is if you start to fold, like in a helix or in the fleeted sheets. And then if you fold again and start to put them together in a big unit, that is tertiary. They then make bonds that holds that shape together. Not only are they just long as primary or twisted like secondary, but now they're folded back together and bonds hold them in shape. That's a tertiary. If you take one or two or more of those and then put them together, then that's quaternary. So it's pretty straightforward and simple, but make sure that you understand those whenever we get ready for the exam.
Do you get a final date on when that next test is going to be?
Same one I told you the other day.
Okay, so we are doing that because I know you were asking us
Monday.
about different options. So.
On Monday.
Okay.
Same, nothing's changed. What we will do, we will finish up with nucleic acids today, get started on the cells. We'll finish up with the cells Wednesday, and then Friday we'll have time to do a reasonable little review for you. Normally I don't do reviews, folks. You guys are lucky. And particularly for those of you coming out of high school or still in high school, you got it made compared to the way that you used to do it whenever I first started doing this. You had 2 exams, the midterm and the final. That was it. They were big exams, and people did well. It was harder on them, but they did well. And the reason I switched is because I think this gives people more time to actually master that material that we're talking about. If you're going to go on in these areas, you do have to master this material. There's certain things that when you come out, you should know. Things, we go back like what's a polymer, what's a monomer, what determines the shape of a protein, were the proteins located? What did they do? Sell, save, restoration? That's the type of thing people are going to expect you to go into next year. Now, to back up just a second, this is 1081. In the spring, you take 1082. 1082 gets away from the cellular level and starts to get into the organism level. We'll start to talk about taxonomy, the different groups of animals, different kinds of plants, how plants function and animal function and things. So it's getting up more to the animal side. As a sophomore, you will have to take cell biology, which is almost what we're doing in 1081, except in much more depth. There won't be any new topics. It will just be the topics we'll go into deeper. Genetics will cover a lot of what's covered in 1081 at the end. And you will start to get to see genetics in a little bit deeper form. And ecology is how the animals all function and work together. Yeah.
Oh, may I ask a few questions about, I have actually a few questions. What exactly did we have to know about the Zwitter ion? Because I was kind of confused of which one is the Zwitter ion. Is that the one that gives up its,
It's on an amino acid, and the whole thing is then called a Zwitter ion. It's just where the hydrogen proton leaves the carboxyl group and goes to the amine, that's it. And
Okay.
basically, that's all you need to know.
Thank you.
Okay. Today, we're going to cover something that can be difficult to conceptualize. There's not an immense amount of material, but this is something you really need to stay focused on, because all of those courses I just talked about, these are going to come into play. This is the stuff that makes up DNA. You can name it different things, just simply based. Some of these things are going to be called nitrogen bases. In other words, they are a little molecule, and the main component of that is going to be some kind of a nitrogen. Adenine is one. If you take that adenine and then you add sugar and phosphate to it, it's called adenosine. Now, adenine is one of the bases that makes up DNA. It's also the base that helps to make up ATP, adenosine triphosphate. So it's adenine when it's in that base because it doesn't have the other stuff added to it. But when you add the sugar, a ribose sugar and a phosphate to it, then it becomes either AMP, adenosine monophosphate, ADP, adenosine diphosphate, or ATP, adenosine triphosphate. And so it's the number of phosphates added to it. But the key to that, remember, is then it's not called adenine triphosphate, it's adenosine. But the base is the same. It depends on what else is added to it to change the molecule. So, let's take a look at these. Once again, you're going to have to draw one of these, so be prepared on these ends. You will have to draw ATP, remember, that I told you you'd have to draw. You're going to have to draw one of these. You're all going to have to draw an amino acid.
So when we're drawing ATP, that would be adenine, right?
Adenosine.
Oh, adenosine, and then attached to the phosphates, or?
3 phosphates attached to it. Yeah.
So we write down adenosine, okay.
You can do a little rectangular box, adenosine, and then 3 circles with a P in it for the phosphates. Remember, I said to draw phosphates, you can just draw the circle and a P. That's done a lot of times in here.
Okay.
And what's the difference between if you see PO4 or OPO3? Still 1 phosphorus and 4 oxygens. Remember, it's the ionized form. When it's ionized, it's OPO3. Remember, it shows you on functional groups. So make sure you know that. I guarantee you, you're gonna be asked those functional groups. You may be given what is COOH. You'd put carboxylic acid, or it may say, write the letters for carboxylic acid, COOH. You need to know those. You need to understand things like that, Zwitter ion in the ionized form, where the ions are moving around. That's all it is. So, look back on those functional groups. Make sure you can understand and draw a dehydration reaction that we looked at. And this will show you one when it comes on. You want to be able to understand that when a dehydration reaction takes place, it puts molecules together. They grow, they get bigger. And to do that, you have to get rid of a water molecule in order to make places for them to bond. Also, remember, catabolic reaction makes a molecule grow. A anabolic reaction, catabolic breaks down big molecules into small ones. Anabolic makes the molecule grow. So if you were to look at this is a dehydration reaction right there. You should be able to draw some form of that just to show the monomers of the water being expelled. Little things like that, okay? Once again, this test should be straightforward. There can be a lot of material, but I do not try to get tricky, folks. Promise you.
I'm sorry, did you say draw the hydrolysis and dehydration, right? That's what you were showing? Draw the hydrolysis
You should be able to
and,
draw either one, either dehydration or hydrolysis.
but that's what you were showing on that slide? Okay, thank you.
Now, we often hear terms about genetic material, DNA, RNA. It refers to the version of the sugar that's attached to that nitrogen base. DNA stands for deoxy. What does that mean? Oxygen removed, right? Ribose sugar. So it's a sugar that's got some oxygen gone. So it's deoxyribonucleic acid. RNA is just ribose. The oxygens are there. Ribose.
Like a sugar, I think. Oh.
These are the two types of nucleic acids. Remember, the big strand itself is called the acid. DNA provides direction for its own replication. Actually code for its own replication.
I'm sorry, what did you say Rebo represented?
It's a sugar, ribose.
Ah, thank you.
Now, here is the key in the way your body works and cells work. DNA holds the codes for proteins. Those proteins will be in a sequence of amino acids. DNA directs the synthesis of messenger RNA, and through mRNA controls protein synthesis. Here is what I want you to remember, and I will ask you this on the exam as well. I'm going to go to another little slide here, and it will make it more clear to you. Messenger RNA is a copy of a gene off of DNA. If we've got DNA in a cell, we can't cut that part out to make a protein from it. It would destroy the DNA. So what we have to do is copy that gene. The copy is called messenger RNA. mRNA. Remember, a gene will code for a protein. That protein will be made in an organelle called the ribosome. The ribosome itself is a kind of RNA. It is called rRNA, ribosomal RNA. So once we got a gene copy, messenger RNA, and it goes to the ribosome where it's going to be made, the only thing that's missing is the amino acids that it needs. And that comes in the third kind of RNA, tRNA, or transfer. It goes out, picks up the amino acids, brings them to the ribosome, where they're then put into order to make the protein. Protein synthesis occurs on ribosomes. Now look up here, follow through with this. This is the nucleus. This is the cell, the inside of a cell. DNA is so fragile, we don't want it out into the middle of the cell where other things are going on. Some of them caustic that can damage DNA. We want it in its own little protective structure. That is the nucleus. This is the DNA, and let's say there is a gene on DNA that we need to make a protein for, insulin, for example. We need to copy the code. So enzymes will immediately go to work and start to copy that. They will make messenger RNA, which is going to be a copy of that gene. That copy must leave the nucleus, and it goes through these openings called nuclear pores, P-O-R-E-S. They leave. So we've made a copy of the gene, and now it leaves. Comes right through the nuclear pore. From the cytoplasm, it's going to go to the ribosome. The ribosome is one of these proteins that is different structures. It's got parts to it. That ribosome will enter in through here. This ribosome is kind of like almost a vice that holds it in place so everything is lined up. It has certain openings in it that will allow transfer RNA to come out into the cytoplasm, find the correct amino acid, and then notice start to put it together. So the copy goes through the ribosome, and it's read. It says to put a proline here. So the proline comes in, the tRNA drops it in place, and it's bonded in a peptide bond, and the chain keeps growing. So you're just simply putting one section of amino acid together at a time through there. These are the amino acids, and this is the growing peptide chain.
So the gosh, what was it? The transfer RNA comes out of the ribosome and it goes into the cytoplasm.
It don't come out of the ribosome. It's in the cytoplasm.
Oh, okay.
And then there will be messengers, chemical messengers, and then when the transfer RNA picks up all these amino acids, it will only let, there is a certain code on this, and it will only let that one amino acid attach to it, nothing else.
So it picks up the amino acids from the mRNA, and only
No, it picks up the amino acids from the cytoplasm.
Okay, from the
And it
cytoplasm.
goes to the mRNA, and the mRNA tells it what order for them to be assembled. We'll go over that in more depth later. The big thing right now, for you to understand, messenger RNA is a copy of the gene. rRNA is the ribosome where the protein's made. And transfer RNA brings the amino acids in from the cytoplasm. That's all you need to remember for now. Notice the nucleic acids, DNA and RNA, are polymers called polynucleotides. The monomer will be the nucleotides. Poly just means many. Each polynucleotide is made up of the monomers called the nucleotides. You'll see what they are. Now notice here, a nucleotide consists of a nitrogen base that we've talked about, a pentose 5-carbon sugar, and one or more phosphate groups. Carbon sugar is ribose. Now, when it's just adenine, for example, It's just called a nitrogen base. When you add these sugars and the phosphate groups to it, then we change the name to a nucleotide. And you'll get to see later, if it doesn't have the phosphate group to it, there's even another name. It's called a nucleoside. But we'll get into that later. Well, here it is. It says the portion of a nucleotide without the phosphate group is called the nucleoside. So if we have a nitrogen base and we add a sugar to it, it's called no longer nitrogen based, but a nucleoside. If we then add the phosphate to that sugar, it becomes a nucleotide. A series of nucleotides put together then make up the polypeptide. Now, here's one of those, notice the nucleoside right here, the base and the sugar. When you add the phosphate group, it becomes nucleotide. You should be able to draw this. One ring that says nitrogen base, one ring that says ribose, and then the phosphate. You can just draw the O and the P. But you should show that a nitrogen base is attached to a sugar, ribose sugar. You don't really have to draw all of the rings and the angles. You can just kind of draw little blocks, square blocks if you want to, and just say nitrogen base, ribose sugar, and then the phosphate. Now look at this nitrogen base right here. In all of these over here, which one looks most like that? Not with all the other stuff attached to it, just the shape of the ring. Top one, right?
Right.
If you take away all the lettering in black, the overall shape of these resembles that one. Those are called pyrimidine bases. The key to a pyrimidine, it only has one carbon ring, like that. The opposite of a pyrimidine is a purine. They have two carbon rings. There are a lot of importance to those. And these will be the sugars they're talking about, whether it's deoxyribose or ribose. You can just call it ribose for now. So we are going to have a phosphate, a sugar, and then some kind of base. That could be either a pyrimidine base with one ring, or it could be a purine base with two rings. Here is the importance of that. These represent those bases that we just looked at. Pyrimidines that have only one ring will be smaller than the purines that have two rings. Thymine, a pyrimidine. Cytosine, a pyrimidine. Look how small they are compared to the others. Purines are bigger. The way this kind of ladder, or whatever you want to call it there, that makes up the helical structure, that is going to be made up of the phosphate and the sugars starting to bond and stack on each other, and that makes the big rail. The phosphate and the sugars make the rail. The bonding that holds DNA together is going to be between these bases. That will be hydrogen bonds. it's important that they're hydrogen bonds because these are bonds we want to be able to break when we have to copy DNA. Now, there is enough space between these rungs for a small pyrimidine and a larger purine to come together and adequately fill in that space. If we tried to use two pyrimidines like adenine and guanine, there would not be enough room. If we used two pyrimidines, they would be too far apart and the bond would not form. So we have a purine and a pyrimidine because when they come together, that fills the space and gives enough room to make the hydrogen bond in between.
So the thymine, right? That's the T. Is a no, not a purine. You said it's a pyrimidine, and then the what is that? Adenine.
Purine.
That's a pure. Okay, that's a purine. Okay, and so the guanine or guanine, would you call it?
Adenine and guanine
Are purines, right?
are the purines.
Oh, okay, it's right there. I apologize.
Up here, cytosine, thymine are pyrimidines. In RNA, thymine is not used. It is substituted for something that works and functions the same, but called uracil. That's only in RNA. In DNA, it's adenine and guanine, thymine and cytosine. In RNA, it's adenine and guanine, cytosine and uracil. So these are the purines. These are the pyrimidines. And if you notice over here, notice these green, this is the base. Notice how it sticks out. This makes up the rail. Up here we get the phosphate and the sugar. Phosphate and the sugar. Notice how they're attached to each other, and they make that ladder, the rung rail structure of DNA, the sides. Those sides are like a ladder, and they hold the nucleotides together, the nitrogen bases. These are the nitrogen bases that are now available to bond to this pair. This is called complementary base pairing because they do actually complement each other. Remember, the nucleoside is just the base and the sugar. When we have the phosphate added to it, the base, the sugar, and the phosphate, then we have the nucleotide. This is what we have just talked about. You can read up on that on your own. Remember, the sugar in DNA is deoxyribose. Oxygen's removed. In RNA, it's just ribose. And then the nucleotide is the nucleoside. Any questions on any of that? It's pretty simple stuff. You just got to go through it and remember it. Yeah.
Okay, so basically when we draw the nucleotide, we'll basically draw what's shown, but I know that we can draw a P with a circle around it simply for the phosphate, correct? And then for the sugar, the pentose, we basically have to draw what's shown, right?
A box with ribose written in it, then a box with adenine or nitrogen base written in it. Remember now the nucleotides are polymers, or they will come together to make polymers. Nucleotide polymers are linked to build a polynucleotide. Adjacent nucleotides are joined by covalent bonds that form between the hydroxyl group on the 3' carbon of one nucleotide and the phosphate on the 5' carbon on the next. Now this is out like in one rung. These are all formed together to make that long rail. Those are covalent bonds. We don't break those ever. We want those nucleotides to stay in the proper order. These links create the backbone of the sugar phosphate units with the nitrogen bases as appendages. If you go back now, that's what they're talking about. These are covalent bonds that form to make that rail. And then that leaves these, they call the nitrogen bases as appendages. sequence of the bases along a DNA or RNA polymer is unique for each gene. RNA molecules usually exist as a single strand. It can be double in some organisms where the primary genetic material is RNA instead of DNA. But in most cases, RNA is just a single strand. DNA molecules have two of the strands spiraling in an imaginary axis, forming a double helix. In the DNA double helix, the two backbones run in opposite directions. Now, these have directions, remember. The hydroxyl end here is called the 3'. The phosphate end is 5'. They go in a specific order. Now, what's going to happen is one strand goes 3 to 5. It flips for the bottom strand, and it will go 3 over here where this 5 is, and go back the other way. And that's why everything lines up accurately, and you have codes for the same bases on both strands. And remember, when we are talking about DNA, it does not matter which strand you're looking at, they both code for the same thing. They are copies. Why do you think we need to have two strands that code for the same thing? When a cell divides and becomes two, one strand goes into each cell, then each strand is copied. So then we wind up having a full strand set of DNA in each cell. That's why we have two. It can also act as a spell check, in a way, for the genes. This is called antiparallel, and they go in different directions. One DNA molecule has many genes. A human being has around 14,000 genes. Thing about it is, we don't know what they all do. A project called the Human Genome Project has identified the genes, but we don't know what all of them do. You can actually go online, it will show you the 23 chromosomes, and it will show you gene X432 or something like that, number given to it, and then off to the side it'll say unknown function. Or it may have one that says, this is the gene produces insulin. Some we know, some we don't. And as we get into the actual structure of DNA later, you'll find there are sections that code and sections that do not, the introns and exons. And here is something else to remember about DNA. Only about 1.5% of all DNA actually codes for a gene. So the DNA is much larger. It's got other functions in that other area that will help it in making proteins and copying itself. So only about 1.5% actually produces codes for genes, but the other part we think does have a function to it. The nitrogenous bases in DNA pair up with and form hydrogen bonds. That's the ones that holds the two rails together between the two nucleotides. And remember, adenine always pairs with thymine. Guanine always with cytosine. If we're talking about RNA, adenine would bond with uracil instead of thymine. Any questions on that one? We will cover how DNA completely replicates. We will cover how every gene is turned into a protein. But that's coming for later. Right now, we're talking about the chemistry of the molecules that will do those things, how they're put together. And once again, this shows the picture that I've been referring to. Notice the three prime and 5 prime. This one goes in that direction. The other one will code for the same amino acids in the same place, coming back the other way. Yeah,
So the 3' and 5' are basically, they're opposites of each other, but they're the same copy, right? Is that,
code for the same amino acid to be in the same place.
but what exactly does the 5 stand for and the 3? I mean, where does it come
It is the
from?
direction in which they're going. Because of the bonding with the sugars and the phosphates in the nucleotide direction. Don't worry about too much what it means right now, just understand the directions and that they do the same thing.
So you can say which direction the 3' is going? And which is going, because,
3' to 5' is considered forward.
oh, okay. That little mark above the 3 means prime?
Folks, we've got people on phones and computers doing everything, having a good time. Just remember, you've got a test coming up. It can be easy, or it can be not so easy. We've got time to do other things, then let's go for it. Mind you. Cells. All of these cells that we're looking at, or we've talked about, they have a generic setup and structure. It's going to be a membrane. Inside of the membrane is going to be a jello-like fluid. It's not going to be watery. It is going to communicate through all parts of the cell, and it will hold the organelles. Organelles are the parts that make up a cell that do different functions that we will see. Notice the cell structure is going to be based upon the cell function. We've got a lot of different kinds of cells that do different things. Nerve cells that can be that long, literally. People think of nerve cells being small, they're not. Muscle cells can be like that, long also. Some cells are microscopic and little round things. All cells are different. They have different functions and different structure, but the basic structure is going to be the same. They're going to have that membrane around them and then the organelles inside them. And remember this term, or this statement. I will ask you, where do cells come from? And your answer should be from other cells. Cells don't just grow on their own. They don't up out of the air. They must come from other cells. One of the things that's been studied a lot in things such as evolution, ancestry, how did life begin? One of the big questions for a long time was, has life popped up one time, or did it pop up multiple times? Did one time it make animals, one time plants, one time bacteria, one time fungi, and it has been found without any doubt one time. That's all life's ever formed. What do you think the odds of life forming are? Think it'd be as much as winning the lottery?
Probably less of a chance of life forming than winning the lottery.
People have studied this for a long time. And how life formed is still a mystery. Nobody can tell you. And you'll hear things on the news, how people have almost in this lab or stuff done that. Nobody can ever even come close to forming life. Never. Not even close. They can put some chemicals together in soups to emulate what the early environment would be like and maybe make some coagulated mass of polypeptides. It's not life. not even close. It's formed one time, and there was one guy, there's one Academy of Science here that's really famous and well-respected, Chicago Academy of Science. And they will meet yearly and talk, and some of these guys, they'll almost get in fist fights, arguing about a lot of this stuff. And one guy calculated what the odds that life could form once. And it came up to be 10 to the 150th power. That would be 10 and 150 zeros. That would be the odds. We don't even have a name for the number that big. And he took into all the different, think of how many different combinations we can get with just 6 numbers on a lottery. What happens if you expand that many, 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 many times? You have to have the environment that's just right. You have to have all the parts put together. Who's gonna make these parts, by the way, where they come from? We're assuming that they're there, but they had to come from someplace. Most of these things are made by, guess what, life cells. But he came up with that 10 to the 150th power, and it's ridiculous. He said that would be the same odds, and I'm taking this from him. This is not me saying this, I'm just relaying what he said in an article, that the odds of life forming on Earth is the same as a tornado hitting Rumpke and making a working 747 airplane out of all the garbage. Just throwing it all together. Or, same odds that you walk outside, take a straw, face the west, throw it up in the air, having the wind catch it, take it all the way around the earth and bring it back and putting it back in your hand from the east after it's traveled around the earth. That's what he said the odds were. For that reason, many people have come up with some ridiculous ideas on how life formed. One of those, some people, one of these guys, now these are people with PhD, thinks that all of us are a science experiment for some little alien kid out in space, that we're just like his little test tube and he's using us. That's where you get things from like, life had to come from outer space. Well, guess what? Even if it did come from outer space, it still had formed, didn't it?
I've heard a theory that we may have all come from Mars.
There's one,
Moved to Earth.
Mars is just Earth at a distance, so still had to form. There's some people say we don't even exist, that we're just like a dream. That's what they say. I don't believe any of it, but I'm just telling you what people will say. And the key to this is life is formed once. All cells come from other cells. They don't just pop up out of the air. Now, you can have a cell and have them branch off into many different directions over billions of years, but they still come from one source. We have never been able to replicate life. One of the good or the most accepted theories at one time was called the warm little pond theory, which is basically where that guy came up with the 10 to 150 power, whereas the earth had water on it. The atmosphere at that time was like basically when life did form. We know when, but it was like a microwave oven. Literally, if you were on the surface of the earth, it would be the same as being inside of a microwave oven. We had no atmosphere blocking out anything, no water vapor in it, nothing. It was just the sun pounding us. It took 1 billion years to develop enough atmosphere for life to start to form, 3.5 billion years ago. Still, the radiation would have been enormous. So people said it must have formed in water. It makes sense. Water helped to act to diffuse the ultraviolet radiation. There's only one big problem with that. Most of the proteins we looked at are hydrophobic. They don't like water and they won't form in water. That's a problem. So I cannot give you the answers. Nobody else can. We don't know scientifically what happened. We do know all cells are related. One cell comes from another cell. Albert Einstein, his theory of relativity is now just called relativity because it's been proven. He said there's more than one universe. How can there be more than one universe? A universe is supposed to contain everything, right? How can there be more than one? Not only did he say there's one, it's got boundaries. He said it's cone-shaped, like an ice cream cone. He said if you traveled across that cone, instead of going around the edges of the cone, if you cut across the middle of it, if you were traveling at the speed of light, you could turn around, see your own self coming, and then shake your own hand. And it's been proven mathematically. I don't understand it, don't pretend to, but that mathematical calculations have been done to show that. And the biggest calculation that he needed, the reason his theory of relativity was called a theory, was because he didn't know how much the Earth weighed. With the advent of satellites, he did come up with a constant of how much the Earth weighed, but he couldn't prove it. Satellites proved it. So now they don't call it theory of relativity, it's just relativity.
What about the atomic theory? So that's still not proven because you
Well,
can't.
not very much proven, yes.
So why is it still called a theory then? I've wondered about that. It's not?
Atomic theory, it just was meant to be more, it was a theory at one time. Einstein proved that real quick. And we've done it. There's no theory about it, it's just a theory of the way things come together. Some things we can't prove totally, but we know they happen because we've seen it. Here's the cell communicating with another cell. These are the endings of a nerve cell attaching to another cell. And if you notice, these little areas right here are where chemicals from that nerve cell are going to be released. That's going to tell that cell do something, contract if it's a muscle cell, for example. So when we look at cells, remember there's going to be all different sizes and shapes. We will look at the microscope to see many of these. Remember, light microscopes are what we use up in the lab. There are different kinds. They're scanning electrons. The most that you're going to get out of a light microscope, full immersion, would be 2,000 power. Can't hardly see anything at that. We use go up to 1,000. Scanning electrons will measure quite a bit more. But they don't actually look at the image in an electron microscope. They actually take the sample and bounce electrons off of it that then come back, hit a screen, and then it draws a picture of it. So, we've talked a lot about the microscope in the labs. You should know what magnification and resolution and contrast is. Most of that we've covered in lab, and that's where you will hear or be tested over. I want to finish with this one. We got a couple of minutes here. When it comes to nerve cells, there are many varieties and different kinds. Notice, we can have a 1 meter length of some nerve and muscle cells, over a yard long. A chicken egg is one of the largest cells, or an ostrich egg. It's a cell. Now, it's a haploid cell. It only has half the chromosome, but it's a cell. Then we can keep going down. Notice in this area where it's yellow, most plant and animal cells are going to be in this range. And don't try to necessarily remember the numbers over here. This is 1 meter. This is 100 micrometers, which would be a tenth of a millimeter right here. That 100 would be a tenth of a milliliter. This is going to be the areas, and notice there are many other things. When we start to get down to the viruses, ribosomes, and some of these proteins, we just don't have a microscope that's going to get down small enough to be able to see them. However, if you look down at the bottom, with the electron microscopes, they can actually see the electrons move around the nucleus of an atom. And it makes a cloud. They never see it in one spot, but it makes a cloud of the fact that it's traveling. So notice in this range, we've got the light microscope. But when we start to get down to these other things, we have to have the electrons. Here's a few pictures. Cells, for example. We can see cells like this when you guys go in and do the labs on the buccal smear. You'll be able to see cells that'll look like this, except they will be blue, and they will be your cheek cells, but you'll be able to see them because they're bigger cells. However, when you start to get to things, these are the cilia on a cell. We can't see those because our microscopes just won't go down that far. And so we're limited. We can see the basic cell walls and structures. We can see the nucleus. We can see the chloroplast in plants. But we are limited when it comes to looking at things like the ribosomes and the other organelles. Now, this talks about the different kinds of scanning and transmissions. If you get a question on the test, it's going to be this. The lights, in a light microscope, it has a light source, just plain light, visible light. An electron microscope, however, uses electrons to bounce off of it. That is the image source, not light, but electrons that hit it, bounce back up to a screen. You can actually get a PhD just in using electron microscopes that are not easy to use, and it takes a lot of knowledge. They actually have an operator's chair, most of them, like a piece of equipment. Wednesday, we will try to finish up with this.